Okay. All right, so you'll see a lot of similarities, I think, between Alan's talk and what I'm going to say here. The emphases are a little bit different. We disagree on one or two things, and that's all good, right? So we're good colleagues and stuff. I took a couple of different other people's graphics over on the left side of this, but they kind of like speak to this integration of molecular and morphological or phenotypic data more broadly. The title is a play on a title of a paper that I wrote in 2005 about Renaissance taxonomy. Both allude to the periods of intellectual advancement in the Western societies through the 14th to 19th centuries. And I would argue that we're at a time when there can be a new enlightenment, a new age of reason, by bringing more data to bear in our approaches to taxonomy. Okay. All right. So for 250 years, right, we practiced systematics by looking at relatively few morphological characteristics that were not quantified, and the simplicity and ambiguity in the characteristics were interpreted subjectively, and it led to essentially a lot of confusion in Scyphozoan systematics. Right. This is not unique to Scyphozoan systematics, right? It's for many of the gelatinous zooplankton. The examples I'll give are Scyphozoans. And the problem with this, right, as that Alan has said, is that the species are where we put all of the other information, right? It's how we organize stuff. It's how we understand life on Earth. It's how we understand life in the plankton and how we interpret it. So we have to do a good job of making the boxes and putting the stuff in the boxes, right? So in around 2000, essentially, we started to introduce quantitative morphometric techniques and molecular data for trying to identify species of Scyphozoans. Not the first to do this in taxa generally, right? But we try to introduce it to this. And it generates really quite remarkable results, right? And so this is an example from Aurelia. You've seen this tree already this morning in part. And because you have suddenly thousands, not a few characters in DNA versus the qualitative morphological data, and because you can have quantitative characters in the morphology and many more of them, you gain a great deal of resolution and a great deal of stability, right? And these things are repeatable because they're quantitative, they're measurable, they're not about subjective interpretation. And for the most part, you will get reliable taxon or OTUs, if you want to call them that, operational taxonomic units, that are morphologically distinguishable most of the time, right? Not all the time, most of the time. Yeah, I will want that. Okay. But not all the time, right? And the reason is that jellyfishes or gelatinous zooplankton, they are simple, right, in terms of their morphology. We can probably go measure many other things about them, other phenotypes of Alan talked about, you know, variety of techniques, physiological things, behavioral things as well, right? And try to put them in these boxes. But for morphology, it is ultimately limited at some point. How limited? Well, there are some couple of like basic mathematical issues here, right? So you essentially need about this many number of characters that are informative and reliably distinguished species to have a fully dichotomous tree, right? But that's not a fully dichotomous tree that is statistically well supported, right? To do that, you need something like this number of characters, right? Reliably informative characters. So where are we with Aurelia? Well, so John Lawley's paper, right, reckons that there are maybe about 28 species currently identifiable of Aurelia. So that would mean for a fully statistically resolved tree, we would need that many characters, right? How many do we have? Well, it's probably something around this in terms of things that we measure, right? But the things that are actually reliably informative, well, there's some variation in these, so maybe you get more, but then a bunch of them are not informative, so you actually end up with fewer actual informative characteristics, right? So our shortfall in morphological data is that for generating a resolved tree, and this for generating a statistically robust resolved tree out of morphological data only. And that's for the medusa, which is relatively morphologically complex, right? If you're trying to identify from Ephiri or polyps, then it's a much harder problem. And this isn't to say that we shouldn't measure these things, right? But that we should not have an expectation that we can resolve taxa from these things. But it's kind of not the point anyway, right? The point is that we can measure these things and then we can start to put them into these more reliable boxes that we have that are taxa. And by doing that, we can learn about the way that they make their living and we can compare the ways that they make their living. So we can identify characteristics that will distinguish some taxa from another, right? It might not give us all the information but we can do that. In generating this information, we also can identify differences between members of the same species that are in different places, right? The key thing here is that we're generating information on variation, right? Variation is the substance of evolution, is the way that things make different livings. And so it starts to generate important questions, right? So simply the act of measurement generates these important questions, such as why do these species differ or not in not only their gross morphology, but also particular traits. And we can put that into its geographic context, which is also its environmental context, right? So then what are the factors that explain trait variation, whether that's genetic or morphological or some other kind of phenotype? Why is it distributed the way it is spatially, environmentally, right? These are the key questions that we're interested in. The key questions will help us answer how things are going to respond to change in the future. An example of this, I've shown this figure before. Still have not written it up, unfortunately, but we'll change that, try to change that soon. Is this information about morphology and genotype tells us something about the way things make a living, about patterns of diversification historically, about functional traits, right? And give us a macroecological, a big picture understanding of Scyphozoan diversity in this case. And so the example that's up here is from Lisa Gomez-Daglio's fantastic work in tropical Eastern Pacific, where she found 25 new species. And what it shows is that rarely do you find a species within the same genus occurring in the same location, right? You almost always find species, the taxa that co-occur are in different genera, different families, right? These are the exceptions for Chrysiorum. And so this suggests that there's some kind of functional exclusion between species, right? This is niche separation, niche diversification. This is not how we used to think about the plankton, right? It's a bunch of things that drift around, they're generalists, right? It's not what plankton is, right? We learn this through this integration of phenotypic and molecular data that allows us to build these reliable structures for boxing up the information into things. All right. Let me give a totally different example of some of the ways of integrating phenotypic and genetic data. So about 5 years ago, this is from Carly Higgins' thesis. About 5 years ago, we happened to be in Palau in marine lakes. This was one of those periods of times when there was not many jellyfish in the lakes, right? There are usually millions. People will go as tourists to go see this phenomenon of many millions of jellyfish in the lake. There weren't that many this particular time. But the interesting thing was that there were some animals that were like normal, zooxanthellae, right? So they have this brown color from zooxanthellae. And there were others that were syntopic in the same lake at the same time, just intermingled with each other, that were white and largely azooxanthellae. Okay. And so we're like, are these things genetically different? Are they behaving differently, right? We know that these mastigious might have a diurnal horizontal migration. And so we're like, let's measure it. So we took some samples for genomics. It turns out that genomically, they're more or less indistinguishable. But if you look at the behavior, the white ones don't migrate as strongly as the golden ones. And so the golden ones, this is a Rose diagram that shows an instantaneous swimming direction. Essentially, the golden ones are oriented towards the sun in the morning and in the afternoon, whereas the white ones are randomly oriented, right? So these are genomically indistinguishable hosts in the same environment at the same time. This is the zooxanthella making the migration of the mastigious, right? That's an amazing thing. Like, how does that do that? Right? And this is just one example of the interaction of a zooxanthella and a cnidarian host. And for cnidarians, right, we know about interactions of zooxanthellae and cnidarians and zooxanthellae primarily from a two-dimensional benthic environment, right, corals. And so what happens when you take that symbiosis and you put it into a much more complex three-dimensional environment where you can modify things with behavior, right? So this is one of the questions we're interested in asking now. But it's not just that behavior, right? There's a long history of evolution of symbioses in Scyphozoans. There are at least 2 independent origins of symbiosis within Scyphozoans. There are more if you go out across the other cnidarians. And some taxa seem to have single symbionts, others seem to have multiple symbionts. Why? Right? We don't know. What's the consequence of that? So there are evolutionary transitions to understand. There are also life history transitions to understand, right? Why do they not have vertical inheritance? What are the benefits of horizontal inheritance of zooxanthellae? And for Mastigius, it has kind of like a traditional-ish life cycle in this context, is that you lose the zooxanthellae when you reproduce. But there are other taxa, such as Cephea, that loses the zooxanthellae essentially around here. Why? Right? What are the benefits? What are the costs of having zooxanthellae at different life history stages? So this is a project that is being partially funded in a way by the Moore Foundation and the Wellcome Sanger Institute. The portion of the project this is paying for essentially is this project we call it Symbiosis in 3D. And they're generating high quality reference genomes for 50 species of cnidarian zooxanthellae pairs, right? So 100 genomes together. And the idea is to build a reference genome so that we can go ask all of these other interesting questions about symbiosis. This is the team that's doing it, right? So Alan had a very similar figure like this at the end of his talk, right? It's a global collaboration. We saw Rebecca Helm with a similar thing yesterday. And it's not possible without this diversity of people working in diversity of places, right? And it's not so much fun without those folk either, right? So, but it's not just the symbiosis, and it's not just the fun, it's not just the reference genome, right? The interesting thing is to build the phylogeny, understand the boxes, and to put all of this other stuff into there, right? Is it symbiotic? How symbiotic is it? Where does it live? What are the environmental conditions there? What's its growth morphology? What are the characteristics of, for example, this like highly engineered, apparently, canal systems? Why does that vary between taxa? What's it doing? Which bits of this are actually adaptive and which are not adaptive, right? There is this great idea about Alan, what's the, anyway, I'll come back to it. All right. Not everything you can measure is adaptive is the basic idea. Spandrels of San Marco, right? Stephen Jay Gould, right? Got it. All right. So, in summary, right, we've got like a huge amount to learn about species distributions, life history, their interactions, the morphology, the physiology. Alan named more, right? Systematics, phylogenetics, taxonomy is the way we organize that information. Molecular and phenotypic data are more powerful together, I will argue, right? The simple observation raises questions, gathering data on it focuses those, it allows us to make hypotheses that we can test and learn stuff from. I think we're at a time of unprecedented opportunity, right, in terms of being able, we've seen lots of great talks here, all right, on new ways to collect data, but it will pass us up if we don't take it. And so I think we need to be active about saying we have lots of ways to collect data. We're here as a community. Like, let's chat and let's work out what collaborations we can make so that we can grab this opportunity and learn a bunch of stuff, have a new enlightenment, blah, blah, blah. All right, so I just pulled out like a half dozen examples of some of the great talks we've seen that cover different aspects of this, right? And it's not all of them. Other people here have talked and like given great talks too about stuff, right? But there's lots going on. So what should our next steps be? And I'm being a little bit more dogmatic about this or whatever, let's stand back with Alan, because I think we should do this, right? And we've kind of like had these conversations before. Not necessarily done a huge amount about them, except individually, but as a community, I think we can do these things, right? So I think we should establish some working groups. Let's chat about that before we leave here. We should then identify some priorities, maybe some low-hanging fruit. We should build and expand our collaborative networks, building on strengths and opportunities, right? Some of us are able to do some things in some places some of the time, right? But together we can probably do a lot more, right? So let's like take those opportunities. We should standardize some approaches. I put Mark Gibbons' name up there because Mark talked to me a couple of years ago about essentially writing a bunch of papers on standardization so we can use same protocols so we can actually integrate the data that we all collect, right? We should be able to, we need to identify and create data structures that will facilitate integration, right? It's not enough just to collect the data. If you're a late stage grad student, early career postdoc, like the world is your oyster, right? Reach out to us if you want to like take a leadership role in some of this stuff. You have like the time and the opportunity to do it. And then we'll need to get some funding, et cetera, blah, blah, blah. But we should make it our goal to report